Alright guys, in this PowerPoint we're going to be talking about genetics. More specifically, we're trying to answer the question, how did I inherit my traits? You can bet it has something to do with this molecule, deoxyribonucleic acid. And by traits, I'm really talking about kind of what we look like and also the things you can't see. Basically all the instructions in our DNA that make us and kind of how those get passed down to us from our parents and their parents pass them down to them. So those are the questions we're trying to answer. So if you take a look at this picture, I think it's fairly obvious something is slightly out of place. It's not the boat, not the shed. No, no. And snow, no, up, there it is. This guy. So this deer here is a really great example of asking a question about how we inherit things. My question is, how did this deer inherit these traits? None of the deer around it are very white. I mean, they have a little bit of white on their tail. But how did two of these deer mate to make a deer like that? And this is what people started asking when odd traits kind of show up. Another picture. This is actually called it's not albino, it's actually called a piebald deer. And piebald deers are actually, they've inherited a mutation that causes them to have this odd color. They're all white. They're also, if you look, the front legs are very short. If you get closer, the face is actually shaped a little different. And there's a couple other abnormalities that you don't normally see on the other deer. And you can kind of see the body shape is different. And that's because this deer inherited this genetic mutation, basically, that causes it to be white and have these traits. So we're going to continue on that and ask a couple more questions. Another picture of the piebald. Alright, and one of our first thinkers back in the 1800s to really start figuring out genetics and laying down some basic principles based on the evidence he found in his experiments was this person, Gregor Mendel. Our objective is to describe what Mendel did and how his experiments led to our understanding of modern genetics, because a lot of his principles still play out in what we know today. So Gregor Mendel was an Austrian monk, and he studied patterns of inheritance. Basically, how our traits get passed down. He used pea plants, and he asked how genes are passed from a parent generation to the offspring. At the time, most people were still running on Aristotle's postulation that simply you are, and all organisms are just a blend of the traits of their parents, kind of like a half and half. We call this blending. Mendel saw some things that defied blending, so he studied them further to try to understand if blending was really true, and he found out that blending is not exactly true. He used pea plants, a great organism to use. They grow fairly easily. They have very distinguishable traits, and you can grow a lot of them and see generation after generation where the traits are going by looking at the characteristics of the parents and the offspring. So we have to know a little bit about plants to understand Mendel's experiments. So let me give you the basics here. Plants contain pollen. That's basically a plant's version of sperm. And plants are found in the female part of the flower. They have both sexual organs on one organism. So plants do make sex cells. Guess what process makes those sex cells? Meiosis. Meiosis occurs in the male and female parts of the plant, and it gives the plant either eggs or pollen. And here's kind of the male parts of the plant, and this is the female part here. And flowers are just to make pollinators and other things go ooh and come over to the plant. Pea plants self-pollinate, so many plants can do this, but pea plants certainly can. Which means the offspring will have the same genes pretty much as the parent, because the plant can actually reproduce by itself. There's only one parent in that case, which makes them a good organism to use for this type of study that Mendel did. True breeding plants are basically plants that produce offspring that are genetically identical, meaning they have the same traits. So if you have parent plants with purple flowers, the offspring will have purple flowers. And every time you mate that offspring or self-pollinate it, it'll produce the same thing, identical traits, that same purple flower. This is how Mendel figured this out by breeding them over and over again. So cross-pollinating is another way that plants will reproduce, and most plants actually prefer this because they get to mix up their genetic information, and really in nature, variation is the key to survival. So cross-pollination is when the male pollen of a plant from the stamen, these little yellow things, goes to the stigma, that little green tip on the female plant. And Mendel controlled this just using a brush and some scissors to control when the plants were able to breed. Since it's a pea plant, it makes peas. He would then plant those peas and they would grow, and he would look at the traits of those offspring plants. So, Mendel's laws. So now we're just looking at how Mendel discovered his laws. So we're basically going to look at his experiments, and we will actually be repeating these experiments in class, but we'll be using a different organism that's also useful in genetic studies called the fruit fly. So, get a couple terms down before we go. Genes and dominance. So, Mendel studied 7 different traits, and a trait is just a specific characteristic. Many traits on us that are very obvious are our eye color, blue, brown, all the different colors, our skin color, our hair color, whether our hair is straight or curly, many traits you can see. So any characteristic, and we typically think of the cosmetic stuff we can see when we think of traits, but it's really all your physical makeup, even the things you cannot see. So he crossed plants with different forms of a trait and studied what happened. Basically, he's like, well, if I take a purple plant and a white plant, or a plant with white flowers, and breed them, what am I going to get? So here's some of the traits he covered, or he studied. He studied the shape, so that's our gene really, the shape of the pod of the pea, the color of the pea, the color of the flower position, the pod shape, the pod color, and the plant height. And I'm going to throw in some new terminology as we go, just try to make the connection to the things I'm pointing at, and we'll see as we go. So these are basically the genes that he studied. So there's a gene in the pea plant for seed shape, for seed color, and those genes vary, and those variations in the genes we call alleles. So you can have an allele for purple flower, or an allele for a white flower. It's just really, an allele is just a type of gene, but the gene itself is flower color. Just like I have a gene for eye color, but the color of my eyes could either be blue or brown depending on what I inherit from each of my parents. And peas are the same way. They have a male and female gene coming to them when they inherit during fertilization. So, parents to offspring. So this is kind of the way that we label our genetics and how Mendel's experiments work. So pay close attention, because we will be reperforming this in our own lab. And it's really not that complicated once you get a good look at it. So the parent generation is called the P generation. Makes sense? Parent P generation. So that's really our first generation. And what's important to know about them is they're purebred, or true breeding. So every time I breed this flower with itself, it always makes white flowers. Every time I breed this flower, it always makes purple flowers. Mendel said, okay, now what happens when I mix them? The offspring from that P generation, after they're crossed, that X means cross, always remember that, we get this F1 generation. It's an older word, but you can think of it as the first family. Family 1 generation from the parents. And that is a bunch of hybrids, because now those true breeds are being crossed, so we call these hybrids, because they're kind of like, if you take 2 dogs that are different breeds and you cross them, you get a hybrid, sort of. And so that's what we call these plants here. They're the hybrids. And you'll see more what that means as we go. So take a look at these experiments. Here's what Mendel saw with his eyes. Mendel, he was looking at many traits, including the height of the plant, which is a gene, is controlled by one gene, apparently. So Mendel crossed a tall plant with a short plant. Both were true breeds. Remember, that's our P1. When he crossed these, he planted the seeds, and lo and behold, all of the offspring were tall in the F1 generation. So Mendel instantly was kind of like, that's odd. What happened to the short genes? Are they still there? Are they not still there? And he had to do a little more testing to figure it out. See if you can guess what he did as I switch here. So Mendel determined that inheritance is determined by factors, those are genes, or DNA. He didn't know that DNA was out there, but we know now. So from one generation to the next, this is where he established his first principle of dominance. So some alleles or some types of genes are dominant while others not. So in the case of tall and short, tall is dominant over short, and we can see that because when you breed a tall and a short plant, all the offspring that inherit any copies of the short genes don't show it. So that must mean that being tall is dominant over being short. So segregation of the F1 generation. All 7 kinds of F1 hybrids to self-pollinate to produce the F2 generation. So now what happens is he's going to take this plant here, he's going to cross it with itself, and he's going to see what happens. And that's going to be called the second family generation, F2. So now if we look at the whole thing, what happens? We get a ratio, right? And that ratio, because remember, Mendel made a lot of plants, and he kind of crunched the numbers, and he got down to this ratio. Every three plants was, in every group of plants, there was a ratio that formed 3 to 1 tall to short, or dominant to recessive. So that's our F2, there's our F1. So Mendel crossed each of these, and we finally see, okay, well, these genes didn't disappear, they actually were hidden for some reason, and now they reappear. Now Mendel is really scratching his head. We don't know what happened here. So he knows that these are dominant. Why did this show back up? How are these genes moving around? He's basically back to that question. How are these genes being inherited? So let's continue on. But big step here. We get our first ratio. So if we look at this now, and we look at kind of the genes of the plants, it's easier to understand what Mendel was seeing. So the short gene had separated from the tall gene, and what basically happened was, when meiosis occurs, if you remember, meiosis makes haploid cells. So if this is our diploid here, we have these 2 sets inherited from the male and female part of this parent plant. Same here. So here's our tall and our dwarf, meaning short. After meiosis, one of those genes segregates or separates and goes into the sex cell. Here's our pollen or our sperm, and here's our egg. So now let's see what happens when they recombine during fertilization. Once they recombine, the gametes, the sperm and the egg, in the F1 generation, we see those 2 copies of the gene, and those are 2 variations of the same gene for height, so we call them alleles. So now the big T represents the tall genes, and the little t represents the short genes. And the physical look, or the traits of this plant, are tall. And we know that now because being tall is dominant over being short. So this plant inherited the tall genes. It has the short genes in it, and it can pass them on. However, it's only showing in its physical traits, or its phenotype, we call that, being tall. So F1s are crossed with themselves. They're going to do the same thing. They make their gametes in their male and female parts. We have that segregation again. The alleles separate because meiosis is going to divide those and randomly put them in one of these sex cells to be passed on to the next generation. So each gamete carries a different form of the height gene, which means a different allele, allowing some of the plants to be short. So if we go on, we'll see. Here's our F2 generation. Guess it. This plant's tall. Well, it says it right there. This plant's tall because it has the dominant copy of the gene. This one's tall. It has the dominant copy of the gene, that big T. And our dwarf, our short one, happened to inherit two of the small copies of the gene. So we end up with the dwarf size because there's no dominant allele in there, or no dominant copy of the gene to make the plant tall. So the law of segregation, alleles are segregated into sex cells. So that's Mendel's next law. He figured out that traits are actually segregated or kind of separated when the plant makes its pollen or its eggs. And that's what we see here. The two T's kind of split and go into each sex cell in that that reduction division that meiosis does. So we look at the pictures. There's our F1. It's all purple, right? Every F1 generation. I mean, he probably maybe had 50 plants or more. Who knows? Somebody knows. But he crossed all of them, and he ends up with this ratio in the F2. And he ends up getting a couple whites in there out of all the purples he sees. And that's because this white inherited the two little t's. These guys most likely inherited a dominant form of the gene and a dominant allele and a recessive allele, and maybe this one inherited two dominant forms of the allele, so it expresses the tall traits as well. A phenotype, think phenotype physical. It's the physical trait is really what we see it as, and it's determined by both the genotype and environmental factors. So, in other words, you can have genes to be the tall plant, but if someone doesn't water you, you're not going to be very tall, or you don't get sun or something like that. The genotype is kind of what we represent the genes with, and that would be for the tall plant having the two big teeth. So that's our genotype there. And we'll get much more familiar with these as we start practicing Mendel's experiments and doing our Punnett squares. So we're going to follow the two different genes through each generation. So remember, this was what Mendel did to understand a little bit more about inheritance and really postulate some big-time information that we still rely on. He started with his parent generation, his F1 and F2, the two families, the first family and second family I call them. And in a dihybrid cross, meaning we're tracking two different genes, we call this a two-factor cross. And we're going to see this is where Mendel described one last principle, and we will be doing dihybrid cross eventually. So independent assortment. So in other words, if you're looking at tall genes and color genes, when these genes separate into the sex cells during meiosis, they get all mixed up, and it's random which cell they go into, and the alleles can actually recombine, or the copies of the gene, independently in the gametes. And we'll see this more when we get to dihybrid crosses, which you don't know much about yet, but we'll get there. So don't worry about that quite yet, but just know that that's his third law, independent assortment. So if we look at independent assortment, remember the you got to really look back to meiosis. Oops. Back to meiosis and see that, remember crossing over in prophase 1, that's what's going to mix up these genes and allow them to independently assort into the sex cell. Notice, here's our 4 copies here, we get 2 because of that reduction division. So in this case, we're looking at 2 genes. We're looking at hair color and eye color, and in this case, you can get any combination, blue with brown, blue eyes, brown hair, blue eyes, red hair, brown eyes, red hair, blue eyes, black hair. Any combination of those genes. They don't travel together. They independently assort and they're also segregating. So Mendel's principles, what do they do for us? We'll be studying this a lot more, especially when we get to our fruit flies, and especially when we start studying human inheritance and the inheritance of really the big thing is genetic disorders. So he defined the modern science of genetics, mainly by actually backing up what he said with data. He determined that he called them factors, but he determined that genes basically are passed from parent to offspring. Um, if two or more forms of a gene or allele exist, one may be dominant over others. So nobody knew about dominance. Remember, everybody was into blending at the time. So he postulated or figured out that, or had evidence basically to support that dominance was occurring. Mendel's principles continued. In sexual reproduction, organisms, each adult has, in sexually reproducing organisms, each adult has two copies of a gene, and that's those alleles I'm talking about. Um, and they segregate when gametes are made. So remember, if you have a gene for plant height, there's our each copy, one there, one there. They're going to segregate into the sex cells, one sperm, one egg, depending on the plant and how it does it. Well, really, it would be two sperm, but depending on the plant. But alleles for different genes segregate independently of one another, meaning they're going to split. And that makes sense because that's what meiosis does to the genetic information. And that's about it. So hopefully that was helpful and good for anyone who needs a little bit more support.